First of all, I would like to thank the organizers of the 16th National Congress of Samir, Dr. Mohammed Belmekki, for allowing me to present today. Today's presentation is on the ocular surface barrier in health and disease and will mainly focus on the disturbances observed in ocular allergies and dry eye disease. We define barrier function as the permeability of the cornea and conjunctival epithelium to water and major solutes, including ions. Loss of barrier may also allow passage of larger entities, such as pathogens. This permeability is determined by the epithelial and subepithelial components. In the corneal epithelium, there are two major pathways that may potentially provide passage of water and solutes. First, epithelial components such as mucin layer and glycocalyx would potentially be a barrier to transcellular transport. Mucins, as you see in the right insert, can be either secretory, like mucins 2, 7, MAC5AC and B, or membrane-bound mucins, like mucins 1, 4, and 16. The lower inserts show the distribution of mucins in the corneal and conjunctival epithelium. Second, there is the paracellular pathway, which is composed of tight junctional complexes consisting of predominantly the proteins occludins, zonula occludens, and claudins. Additionally, a relatively acellular epithelial basement membrane serves as an important barrier in the cornea. This slide shows the location of junctional complexes within the corneal epithelium, with only the tight junctions apically located. Please also note that galectin 3 stabilizes the transmembrane mucins over the epithelium. Navigating through the molecular structures, we see that ZO1, 2, or 3, claudins and occludins are the major proteins making up the tight junctions, while E-cadherin and catenins make up the adherent junctions. These proteins usually are the main subject of research when conducting studies on epithelial barrier function. The study of barrier function is important with applications in clinical practice and research because the barrier plays a role in toxicity and environmental studies. It is a major functional outcome in ocular surface inflammation and wound healing research, for example, in dry eye related studies. The epithelial barrier has to be overcome for efficient drug delivery to deeper structures of the eye. Finally, the development of junctional complexes is an important parameter for the differentiation and function of artificial corneal constructs in tissue engineering. This slide presents methodologies available to investigate the barrier function. Amongst these, vital stainings of the ocular surface, fluorophotometry, anterior segment OCT, and confocal microscopy are the ones we are all familiar with in clinical practice. My research in ocular surface disease usually tackled with goblet cells, which I believe have numerous important functions, such as controlling inflammatory responses, delivering antigens to antigen presenting cells, receive and send nerve impulses, interact with keratocytes, and perform immunomodulation in addition to lubrication and wrapping and removing of debris from the ocular surface through secretory mucins. In allergy or dry eye disease, the barrier is disrupted with loss of goblet cells, loss of tight junctions, increased apoptosis, and inflammatory cell infiltration. Now, at our practices in Japan, we're lucky to have many ocular allergy patients, and thus my prior allergy research was conducted almost thoroughly in human clinical trials. The first clinical study I published on ocular allergy was 22 years ago, but still remains the largest series on the ocular surface disease in atopic keratoconjunctivitis. Now, this study confirmed the presence of superficial punctate keratopathy and epithelial defects and dry eye disease in a vast majority of patients. And this slide demonstrates a higher extent of corneal epithelial disease in AKC patients with either short BUT or aqueous deficiency type of dry eyes. The next step was to clarify the pathogenesis of the ocular surface disease in AKC, and that we attempted in two consecutive studies we published in Clinical Experimental Allergy and Allergy journals. First, we noted that eyes with significant epithelial disease had significantly higher staining scores compared to eyes with insignificant epitheliopathy and controls. Immunohistochemistry stainings of conjunctival impression cytology samples for mucins 1, 2, and 4 showed abundant staining in patients and inflammatory cell infiltrates in the epithelium, the quantification of which also revealed significantly higher numbers in AKC patients with significant epitheliopathy. The real-time RT-PCR for mucins 1, 2, and 4 messenger RNA expressions showed significantly higher messenger RNA expressions in patients with advanced epitheliopathy. The findings from this study urged us to investigate MUC5AC and 16 in atopic keratoconjunctivitis in the report I just mentioned. AKC patients again had significantly higher vital staining scores, lower goblet cell densities, and higher inflammatory cell counts compared to healthy controls. Impression cytology showed scanty MUC5AC but abundant mucin 16 staining in AKC compared to controls. And there were intense inflammatory cells within the epithelium. Real-time RT-PCR showed significantly higher mucin 16 and lower MUC5AC messenger RNA expressions in atopic keratoconjunctivitis compared to control subjects. These mucin changes showed also significant correlation with tear functions, vital staining scores, uh, and goblet cell densities, reflecting correlations with ocular surface disease severity. It is our belief that allergen-related peptidases, inflammatory and chemical mediators released, and later on, mechanical friction from the papillae contribute to the ocular surface disease in ocular allergy. When the barrier is disrupted, allergens gain access to antigen-presenting cells and dendritic cells, which release certain cytokines and chemokines, causing differentiation of Th2 cells, and a part of the inflammatory cascade is mediated by Th1 cells as well. Especially interleukins 4, 5, and 13 stimulate fibroblasts to release periostin, including inducing papillae formations. Interleukin 4 also stimulates B cells, which release immunoglobulin E, that induces mast cells to release ICAM1 and interferon gamma, causing further eosinophil, basophil, and neutrophil recruitment and release of epithelial toxic mediators. These all collectively contribute to epithelial breakdown and further barrier problems. Now, I would like to summarize our experience of the ocular surface disease in an aging mouse model of dry eye disease from the SOD1 knockout mice. The knockout induces accumulation of reactive oxygen species, which stimulates NFK kappa B pathway in the lacrimal and meibomian glands and conjunctiva. For the purposes of today's presentation, I will summarize the conjunctival epithelial changes briefly. Now, we observed a significant age-related decline of goblet cell numbers in the SOD knockout mice. Now, as the left insert of this slide shows you, there was an intense or increased DNA stress damage shown by 8 OHDG staining with aging in the SOD1 knockout mice, and note the abundant dark brown nuclear staining, reflecting an increased staining with aging. We observed the same pattern for the late phase lipid oxidative marker 4-HNE, which stains the membranes and cytoplasm with an increased intensity from 10 to 50 weeks. Both mucins 1 and MAC5AC showed decreased staining with aging to an extent more than the wild-type mice. The immunohistochemistry results were substantiated with real-time RT-PCR results as well. Now, ultrastructurally, the conjunctival epithelium in the aging model mouse showed loss of microvilli, weakening or loss of intercellular cohesion, increased cell detachment, and loss of goblet cells in contrast to the abundant microvilli and plump goblet cells filled with mucin 5AC packets in the wild-type mice. The SOD1 knockout mice had mitochondrial disorders, which we believe resulted in the depletion of energy sources of the epithelial cells to function properly, including release of mucins. A rescue study with a mucin secretagogue having anti-inflammatory properties revealed improvement of vital stainings. Likewise, improvement of beta-catenin messenger RNA expressions in conjunctival epithelium with treatment was observed, and this was also evidenced by the immunohistochemistry stainings. There was a concomitant improvement of ZO1 messenger RNA expressions, which also was evidenced by the denser stainings after treatment in immunohistochemistry. Ultrastructurally, treatment with the mucin secretagogue or the rescue agent also resulted in the reconstitution of microvilli and plicae in the ocular surface epithelium. It is our hypothesis that accumulation of reactive oxygen species causes oxidation of DNA, lipid, and proteins, causing cell damage in meibomian and lacrimal glands and the conjunctiva. The accumulation triggers NF kappa B pathway, which causes elevation of tear and tissue levels of TNF alpha and interleukin 6, which invite fibrosis and apoptosis, and release of MMPs from fibroblasts, which disturb the barrier. Reactive oxygen species, and especially the lipid and nuclear oxidation, is associated with mitochondrial damage, causing decreased energy resources for lipid, tear, and mucin secretions. The resultant dry eye disease further adds to the epithelial barrier damage. I thank you all for joining in the lecture today, and thank you again for inviting me to present and for your patience.